Good afternoon. I am Patrice Domingo and I am here to report for the post-laboratory questions. So for the first question, what reagents are used to extract DNA and RNA? So for the RNA, it's sodium hydroxide and ethanol with concentrated hydrochloric acid, and for DNA, it's sodium chloride, ethanol, and detergent. For the second question, do you expect to get the same results on the qualitative test of both DNA and RNA? And explain. No, however, the results in the two experiments should be alike because they yield similar hydrolysate. All tests except Benedict's test yield the same results. Benedict's test has different results because the sugar in DNA is 2-deoxy-D-ribose. RNA, on the other hand, has D-ribose sugar. For the third question, what specifically detect ribose and 2-deoxyribose? Discuss the underlying principles involved. So for this experiment, Benedict's test can specifically detect both ribose and 2-deoxyribose. This is a reduction test that detects reducing sugar. It needs oxidizing agents such as copper sulfate, trisodium citrate, and sodium carbonate. The last two agents are used to maintain the stability of the test, while the copper sulfate will be reduced to copper oxide. Benedict's reagent precipitates change from blue to yellow, green, or brick red depending on the concentration. Also, cyclic sugar is reduced to linear aldehyde. For the fourth question, which is more susceptible to hydrolysis, DNA or RNA? The nucleic acids DNA and RNA are considered as essential in all known forms of life. In order to distinguish them, the DNA contains 2-deoxy-D-ribose sugar, while the RNA consists D-ribose sugar. The ribose sugar in RNA has a hydroxyl group placed in 2' position, making it more susceptible to hydrolysis. In effect, the RNA is more unstable than the DNA.
Good afternoon, Doc and classmates. I'm Trixie Cruz, so I'm going to continue the report. So what other samples can be used to isolate RNA and DNA? RNA and DNA can be isolated from any living or dead organism. Common sources for DNA isolation include blood, hair, bones, nails, tissue, buccal or cheek swabs, bacteria, animal tissues, or plants. What is the purpose of grinding yeast with sun in experiment 6 using a sample with NaCl and detergent in experiment 7, maintaining a cold ethanol in experiment 7. So grinding yeast with sun. Cells are lysed by grinding the yeast with sun. Experiment 6. Cell lysis is a method where the cell wall is broken down in order to release intracellular materials such as DNA, RNA, protein, or organelle from a cell. Yeast cells are grinded with sand and squeezed through a filter under high pressure to obtain a clear cell extract. This procedure ensured that the extract was free of intact cells. Using a sample with NaCl and detergent, NaCl covers the negative phosphate ends of DNA, which lets the ends to come closer so the DNA can precipitate out of a cold alcohol solution. The detergent, on the other hand, causes the cell membrane to break down by dissolving the lipids and proteins of the cell and disrupting the bonds that hold the cell membrane together. Maintaining a cold ethanol. The ethanol is an important chemical used in experiment 7 since it can greatly help in increasing the DNA yield. A cold ethanol can give a good quality DNA by reducing the temperature in the precipitation process. Moreover, maintaining a cold ethanol reduces the temperature in order to prevent and protect the DNA from enzyme activities that can lead to damaging the DNA. A specific example is the DNase, which undergoes DNA degradation. Next is, aside from detection of protein, what is the purpose of performing the Biuret test? Ideally, should it be positive or negative result? Explain. Biuret test is also used to determine if the nucleic acid is RNA or DNA. RNA yields purple precipitate results is a positive result in the Burette test. DNA, on the other hand, yields a blue precipitate result, which is a negative result in the Burette's test. RNA shows positive results because amino acids are bonded with peptide bonds when they are attached to the mRNA. That's all. Thank you.